To introduce our final speaker today, which is Marcela Radtke. She is a doctoral candidate from UC Davis. Marcela is studying nutritional biology with an emphasis in cognitive neuroscience. Her research focuses on the physiological and cognitive consequences of food insecurity in a college student population. Marcela worked on a systematic review exploring the validity of the Veggie meter against dietary measures and blood biomarkers, and assisted with the development of the standardized protocol for the use of Veggie meter in the research setting. Marcela is pursuing a PhD MD degree to merge nutrition research and clinical practice in underrepresented populations through analyzing diet quality and health-related biomarkers with the goal of expanding and improving food access resources. So with that, I will turn it over to Marcela to share your screen.
All right. Hi, everyone. I'm so excited to be here and finally be meeting a lot of the people who I've worked with over the last couple of years, but I've never met in person. So it's so exciting to hear about all of the research that's being done. And I think it lends itself really well to end with the kind of development of the recommendation paper for the use of the Veggie meter, specifically in the research setting. And then I'll wrap up with sharing a little bit about some of the research that is going on here at UC Davis with the Veggie meter as well. So I wanted to start with kind of the impetus for why we thought the standardization of the use of the Veggie meter was warranted. So in 2020, we published a systematic review on criterion related validity of different devices for spectroscopy-based skin carotenoid measurements as a proxy for fruit and vegetable intake. So we saw spectrophotometers as well as Raman resonance spectroscopy in addition to the Veggie meter. And what we saw is in 2020, there were really only 2 papers that met the criteria for this systematic review using the Veggie meter. And even within those 2 papers, we saw that the methodology behind kind of the technique of implementing the Veggie meter was different between those 2 papers. And so this really provided insight into the goals that we were looking for in standardizing the protocol for the Veggie meter. The first goal was really to allow for more generalizability across studies. So looking at different data that were being produced from using the Veggie meter, if the technique being used was different, it's hard to be able to say if those findings are comparable. The second goal was to hopefully establish this standardized protocol to develop a universal skin carotenoid data repository. So like Rachel mentioned in the beginning, we really are hoping to develop and establish recommended ranges for skin carotenoid scores within different populations, whether this is based on different age groups, BMI classifications, and now potentially maybe seeing race and ethnicity play a role in these recommended ranges. And then lastly, one of the major goals of the development of this protocol was to hopefully be able to establish some sort of numerical value between skin carotenoid scores and an approximate number of servings of fruits and vegetables. So if you're looking at an individual pre to post over the course of, let's say, 30 days, and their score drops by 50 points on the Veggie meter, what does that actually mean in terms of behavioral changes in regards to dietary intake? And so with that, this kind of really provided the insight into developing this protocol, which many of the co-authors are here today on this call. And this protocol was published in Current Developments in Nutrition just this last year in August of 2021. And so the development of the protocol really touched on 5 main areas. The first was we wanted to establish current practices among researchers that were currently using the Veggie meter. We then wanted to review the existing literature of all published research and abstracts. With that, we wanted to be able to identify some areas of discrepancy and specific questions, and then go directly to the Veggie meter creators to ask for some guidance in terms of developing a standardized operating procedure for research purposes. After the standardized operating procedure was developed, we wanted to distribute a draft to researchers for user-reported acceptability. If no one was willing to implement the SOP, it wouldn't necessarily be helpful. So we wanted insight from all of you here today to kind of add to the SOP or modify to be used in the future. And then lastly, I think it's important to note that this SOP was really just a starting place. We know that the more research that's being done on this topic, the more we'll be able to strengthen this SOP. And so determining areas for future research were also very important. And so I'm going to go through each one of these steps in a little bit more detail. So to begin, the assessment of current practices was conducted by sending out a survey to the Veggie Meter listserv in April of 2020. A total of 19 respondents completed the survey, which is a response rate of about 35%. As you can see here in the red box, there was a lot of discrepancy in terms of the number of individuals using the Veggie Meter in different ways. So we saw discrepancies in the calibration technique, as well as the anatomical site selection and preparation. And so with that, we really decided, wow, this device is being used in a lot of different ways. We have a need to develop an SOP. And so we moved forward with just that. Next was conducting a review of the literature. So all publications that were published prior to 2021 were reviewed for methodological procedures when using the Veggie Meter. And with that, we saw that many different papers had different techniques when using the Veggie Meter to collect data. But what we also saw was that many of the articles that were published using the Veggie Meter didn't necessarily report on some of the specifics behind how it was being used. And now this isn't surprising because the Veggie Meter does come with an operating manual. But as we've seen with some of the different versions of the Veggie Meter, the operating manual actually did have some differences there. And so it's not surprising that some of the techniques were a little bit different. And so with that, we took the current practices and the review of the literature and really developed the SOP in 4 main topic areas. The first was regarding the device setup and calibration. So we saw that individuals were calibrating the device at different times, using different techniques. And so the recommendation is now that calibration should occur at the beginning of every use and at a minimum of every 2 hours after continuous use. Ideally, the 1 hour mark is preferred, but we also understand that conducting data collection in public health settings, in school sites, obviously time is a constraint. So at least every 2 hours, the device should be calibrated. The second area was measurement preparation and anatomical site selection. And so after extensive literature review, we decided that the non-dominant ring finger is the preferred finger for the Veggie Meter score. Now, this does contradict what is in the operating manual. And the reason why the non-dominant ring finger is selected is because both the vasculature and musculature within the non-dominant hand tends to be a lot less, meaning that hemoglobin and oxyhemoglobin concentrations are a lot lower in the non-dominant hand. And we know that hemoglobin and oxyhemoglobin can interfere with carotenoid detection. And so to hopefully limit some of that inaccurate detection, we advise the non-dominant hand to be used. And then in terms of the ring finger, studies have shown that the thickness of skin on the ring finger is significantly less than that of the index finger. And so for the penetration of light to be a little bit easier, and again, the callousing and the other sorts of contaminants that are often more prevalent on the index finger is one of the reasons that the ring finger should be selected whenever possible. And then in terms of the measurement preparation, so what should we be doing prior to conducting skin carotenoid score measurements? Ideally, washing hands with soap and water is the preferred measurement for a variety of reasons. First is that it's actually able to remove some of the chromophores that may be confounding the measurements. Something like hand sanitizer or an alcohol prep pad may just move the other colorful compounds or contaminants around the hand without actually removing them. And so they may still be in the detection zone. Again, we understand that not all measurement facilities have a hand washing station. So this is obviously to be used whenever possible and when hand washing is available. The third area is data collection and interpretation. And so the recommendation is that triplicate measurements should be collected whenever possible. This average of 3 measurements really helps to reduce some of the variability in terms of sometimes with the placement of the finger, the light can hit at a different spot on the finger and cause a different measurement to be recorded. And so by having 3, you're reducing that variation. Again, the triplicate measure does take a little bit more time. It takes about 90 or so seconds for 3 measurements versus about 30 for 1. So again, understanding that this does impact time constraints. And then the last area really lends itself to the development of the data repository, so the documentation of different individual level characteristics. And so our recommendation is to record age, biological sex, BMI, smoking status, supplement use, and diagnosed chronic disease whenever possible. So again, we know that these factors all have the potential to impact skin carotenoid scores. And so in developing almost a systematic way of collecting that individual data, when possible, all of these measures should be recorded to look at how these ranges shift based on different individual characteristics. And so after the SOP draft was developed, we sent it out via the Veggie meter listserv for qualitative and quantitative feedback on the SOP. So the qualitative feedback that we received was used to modify the SOP. And then we asked researchers to indicate whether or not they would be willing to implement this new SOP in future research efforts. What we found was that a majority of researchers were willing to implement this standardized protocol to really streamline practices moving forward. And then after the user reported acceptability was collected, it really provided insight into areas that future validation research should be conducted. And Virginia touched on a couple of these as well. But we did hear directly from the creators that environmental conditions can impact skin carotenoid scores. So temperature, light exposure, if you're in a brightly lit room or outside, as well as humidity, could all potentially impact Veggie meter outcomes. Now, we don't really understand how or to what extent these different environmental conditions impact outcomes, but it's definitely an area for future research. And our recommendation states that if you are collecting data outdoors, to ideally record both the temperature and whether or not you're under some sort of covered area is kind of a starting point for this area. The next is differences in measurement site preparation. So if hand washing with soap and water is not available, what is our next best option? Is it comparing that to something using hand sanitizer or alcohol prep pads? And I believe Alicia Redolph is potentially looking into this question. Additionally, differences in anatomical site selection. So we know that the non-dominant ring finger, based on physiological values, may be the best option, but what and how do all of the different fingers provide different scores? And so there was an abstract published in 2019 on a pilot study that looked at this, but it was only done in 2 individuals. And so we really need to look further into the magnitude of change that we see in different fingers and different hands. And then lastly, how do the differences in confounding variables and individual level data collected differ? So we know that BMI impacts skin carotenoid scores. Based on Stephanie's work, we may see that race and ethnicity might play a role, as well as biological sex with Virginia's work. But to what extent? Are these enough variable differences that they should be incorporated into statistical analysis? How does this result in changes to potentially the development of the data repository? So really looking into how the different confounding variables may play a role as well.